So I know there's a lot of newfound interest in fungi, people reclaiming that relationship to fungi. There's a lot of community organizations, community gardens, parks that are considering mushroom programming and inoculating their soils and trees and grounds with fungi, their beds with fungi. And so this is like, especially to the Bronx, this is like my love letter to you with regards to fungi. So I hope today you can walk away with a really solid foundation, understanding what is mycology and mushrooms. But then we're going to actually explore that not through the lens of foraging, but today we're going to focus on that relationship through cultivation. And again, this presentation is special for the Bronx River Foodway to build capacity and to sort of give them a primer on what are the considerations as you sort of introduce new life and organisms into your landscape that may or may not be native or may or may not be acclimated to this environment. So my intention here with this presentation is, as a society, as we sort of artificially nurture and cultivate these green spaces, how do we do it ethically? How do we do it intentionally? What is the relationship between the urban environment and the forest and wild environment that's here in the Bronx? And how do we sort of merge the two? So that is sort of the overarching thread to the whole presentation. So a lot of the pictures that I've selected are actually mushrooms that are found in the wild, but we've actually learned how to cultivate. So there's just a special highlight on that to discuss. Okay, so the title is Mushroom Cultivation from Garden to Forest, but also consider forest to garden. So this whole relationship. You can go to the next slide. Okay, so my name is Sneha, but I like to work under the moniker and name Kali Mushrooms. I like to do that because the mycological space is extremely scientific, but I really want to add my own sort of nuanced cultural heritage and perspective and lens to my practice. So Kali is the goddess of death and rebirth from the Hindu pantheon. And similar to mushrooms, people are very afraid of her, but she's essential, right? She sort of allows for the cycle of life. I also like to go by Kali mushrooms when I sign off on things and not Sneha, as sort of a practice of indigenous cultures where they don't take ownership of the work that they create because it's all informed by community and inspired by community. So to me, then Kali mushrooms rather than Sneha Ganguly, it becomes something bigger than myself, something that's collective and cultural and almost a knowledge base or an intelligence that's beyond me, right? So that's sort of my intention of naming my practice, Kali mushrooms. So I want to preface and say I'm not a farmer. I'm not, even though I have sort of a very business savviness, I'm not very commercial in my dealings with fungi. So I come at this topic as an artist. So I'm very experimental, and I sort of explore it in my own way using my own language. Just adding that. And what I am interested in is when I go out into the forest and I forage for fungi, I'm actually not as interested in mushrooms that are edible or medicinal. I like to study fungi that give you pigments and materials. So the colors that you see on the table or the paper that you see on the table, that's sort of what I'm interested in. So it's a whole other niche of cultivation and identification that also exists. So you guys are sort of interested in that pathway. And aesthetically, this is some of my artworks. On the right is one of my first mushroom paper pieces. All the paper is 100% made out of foraged wild mushrooms. All the colors are originally authentically expressing the color of that mushroom that I use, and all the pigments are natural. So the black is wood ash, and that's part of my practice, being in deep relationship with the forest. I just love that the forest bestows to me all the materials that I need. And then as I learn things, as I'm inspired by things, I express them, right? And I also like to marry it with my heritage of Indian traditions of sari and textile science and printing. So I also do some silk painting with mushroom dyes. So that's how the mushrooms. And then next, another group that I've helped found it called Queerdom Fungi Community. It's across the United States, and again, the mycological space in North America, very kind of explicitly, is still predominantly white. It lacks a lot of cultural diversity. A lot of times when you enter these mycological spaces, you're only the one person of color. And it's, for us, it was important to sort of create a container of our own, and to value not just scientific knowledge, but cultural knowledge. And so this is what POC Fungi community is all about. Again, another mushroom paper piece and a painting that I did for their annual gathering. So 2 other things that I'm involved in. I am the New York Mycological Society's Fungus Festival coordinator. I don't know if you guys went to Fungus Festival last summer or last fall, so I do a lot of community outreach, connecting with community organizations, similar to what I'm doing with you, just sort of introducing them, welcoming them, orienting them, priming them, so that as we all go out into the forest, we sort of have some shared ethics. And yeah, so another group and community I'm a part of is called Cornell Small Farms, and the program is called Community Mushroom Education, or Educators. It's a cohort of community educators and organizers that want sort of a background in fungi, and they want to take that information to their communities, just like Nathan is. So this is what we do. We work with educators and try to come up with accessible ways for them to teach their communities. Okay, so you don't have to go back to the last slide. You'll see a lot of community organizing in my photos. And so that is a direct reflection of the things that the mushrooms teach us. You'll soon find out whether it's foraging from wild mushrooms and figuring out what's poisonous and safe and edible, all the way to mushroom cultivation that requires many stages and is extremely laborious. You'll find that this mushroom, communing around mushrooms is inherently a community activity. It's really something we can't do alone safely. So I just love that, just a theme. So let's begin. We're going to get into sort of the 101 of fungi. And here you have the tree of life. And I also like to call the quote unquote mushroom kingdom the mushroom queerdom, because we find that mushrooms are really unruly. And they're kind of lawless, and they sort of challenge your preconceived notions of the binary. There is a fungi that has, I think, 24 or 26,000 sexes, right? And so it just totally, challenges all the conceptions that we have. So inspired by that, and also inspired by an academic by the name of Patty Cation, who sort of regards mycology as a queer science, I title my tree of life fungal queer. You'll see, okay, so for those of you who don't know, mycology is the study of fungi and mushrooms. And you'll see on this tree of life, all the way at the bottom, that fungi in animals, that's us, we are directly related to each other. They are our closest ancestors and cousins on the tree of life, which is amazing. And we'll find out soon why. So one of the things, and I just want to connect it to Robin's meditation and breath work and sort of this, becoming one with the fungi almost. So one thing that makes us similar is that both fungi and humans breathe in oxygen and respire CO2. We are also heterotrophic, which means that unlike plants, we can't just like live off the sun, right? We have to consume other organisms for fuel and energy. The one difference though is as animals and humans, we consume our food, which then goes down our digestive tract into our stomachs. And then the enzymes in our stomachs break down our food, right? Alternatively, what fungi do is they use this method called absorption, and they actually exude the enzyme into the environment. They'll break down the substrate or food, and then they reabsorb the enzymes back, and they sort of eat and move and eat and move all at once. So their stomach is outside of themselves, which is really like, I don't know what that means. Something else that's definitive of fungi is that their cell walls are made of chitin, not so much cellulose like plants are. It's a really hardy polymer, and if you touch my paper, that is a sheet of chitin, so you can see it's quite strong. And then last but not least, in terms of fungi, not all fungi create fruiting bodies. So some fungi produce mushrooms. All mushrooms are fungi, but not all fungi are mushrooms. But the fungal queerdom also includes yeast and mold. In your sourdough bread, your beer, literally thanks to fungi. Even though we're disconnected to fungi and sort of reviving this relationship, they are embedded in our culture and all around us. So that is the fungal queerdom. So I want to go over the mushroom life cycle with you. And this is really important to understand as we explore cultivation, because we'll see that this life cycle in the wild is what we try to artificially produce.
I didn't share earlier, but one of the reasons I got into mushrooms was after learning about mycelium.
Yeah, we'll talk about that a little bit. So let's begin with the mushroom fruiting body. Again, to remind you, not all fungi produce fruiting bodies, a fraction of them do. But for these mushrooms, so what the mushroom is the mycelium's reproductive organ that literally fruits out of the ground, and its purpose or mission in life is to grow as big and beautiful as possible and to spread as many spores as possible, right? So you'll see this is a gilt mushroom, probably an oyster mushroom, and within the gills are where the spores are. So it's a spore-bearing fruiting body, and when it rains or there's a gust of wind or some kind of elemental thing that sort of triggers the spores to release from the gills or from the fungi, they then get spread into the environment. They then travel by water, by air, by other mechanisms to then finally land somewhere where all the conditions are perfect and they can pair bond, or maybe they're asexual and they can fruit themselves and reproduce. They have all the sustenance and the environmental inputs that they need to grow. They're very intelligent that way. So once the spores are released, they then germinate, right? They germinate individually and they create what they call these hyphal strands, right? Then similar to us, and not probably for all fungi, they kind of pair bond together. And what happens then is instead of having one nuclei in the cell, you have two nuclei, right? And that gives the mycelium the potential to fruit and reproduce. So basically, the thread-like strands are hyphae. When they become a network, a larger sort of mat, they are called mycelium. This mycelium, as you know, is basically the mushroom moving through the substrate, which is its food, breaking it down, eating it, decomposing it. And once the substrate, let's say a log or a leaf, whatever it's eating, is completely, quote unquote, colonized or myceliated, it then is ready to fruit. First, you see a primordia, you'll just see sort of it like bursting through the threshold, whether the ground or the wood or whatever it is. And then as there is more humidity and water and airflow and all those sort of environmental conditions, it will grow big and beautiful. They grow quite quickly, especially the ones that are a little bit more ephemeral and have a little bit more water. They grow quicker. And the ones that are like reishi mushrooms are conkier, they're harder, they're actually much slow growing. And they just don't have as much water, they're just more fibrous. So that is the mushroom life cycle. Okay, so this is sort of a little bit more intermediary, but I want to just make a mention of it because I think it's going to be relevant later for your understanding. There are largely two classifications of fungi, Basidiomycetes and Ascomycetes. And what I want to say about them structurally under the microscope, they're different. So Basidiomycete fungi have their spores sort of on the outside on spokes, and they're sort of ready to be released when there's a drop of rain or a gust of wind. However, an Ascomycete fungi has the spores inside the asci, right? So it's encased. And it doesn't get triggered or it doesn't get dispersed from the fungi the way that this reproduces is some insect comes, eats it, and then it reproduces it through its dung. Or it has other mechanisms of reproduction that are sort of, need some kind of symbiotic relationship with something else. And we'll see why that's relevant in just a couple slides. Okay. Yes.
Regarding Ascomycetes, is it only like a symbiotic relationship, not parasitic, or
It could be. Yeah, you're totally right. And we'll get that into this right in the next slide. So fungi have different ecological roles, and these are largely the three roles. We have the mycorrhizal fungi form symbiotic relationships with plants and trees. You have saprophytic fungi, and those mushrooms decompose dead organic matter. These are also the fungi that we learn to grow because we can feed them. And then we have the parasitic fungi. So this is a cordyceps mushroom that has parasitized an ant. We've also, I don't know, you take the cordyceps tincture. We have learned to cultivate one of its cousins, the Cordyceps militaris, that can be found in Pennsylvania. So parasitic fungi feed on living organisms to kind of take control of their host. However, I just want to tell you, mushrooms are rule breaking. They are, so some mushrooms in some relationships, they might begin mycorrhizal or they might begin a certain way, but maybe they end parasitically. So a mushroom's relationship to all the different things that it's connected to might be different. So what might be poisonous to us is really nutritious or medicinal to something else. So just consider your vantage point and what this means. It's not so easy to classify in structure. Go back once. Just want to point out again the photographs. I have picked a morel mushroom, which is an Ascomycete fungi, but it's a mycorrhizal fungi that is in relationship with the roots of trees. I picked this mushroom because it's a really sought after mushroom that's very hard to find, but we are slowly learning how to cultivate it. So it's a mushroom that not too long ago you couldn't find in the market, but we have observed it enough, we've experimented with it enough, we've learned its needs and its kind of life cycle to know how to food it. So again, yeah, cordyceps we've learned how to cultivate, and then this is actually, even though it's orange looking, this is also related to the turkey tail mushroom. It's in the same family. I'm also signaling, well, we know how to grow turkey tails and we know what they like to eat. Well, could we then learn how to feed its cousin that isn't in our sort of food chain yet, but exists? Next, please. Okay, so once again, the mushroom is the fruiting body of the mycelium. It's the reproductive organ, and as you know, mushrooms come in all different shapes and sizes and colors and smells and all the sensory things. These are three really common forms that you'll find. So you will find mushrooms with pores. This is an ash tree bolete. You'll find toothed fungi. So a lion's mane is a tooth fungi. You'll see these sort of teeth-like, spoke-like structures. And then the field mushroom. But there's more forms than this. There's jelly fungi, there's club fungi, there's tubers underground. So this is just three really common ones. Okay, so as we were planning this class, we were trying to discuss what kind of information to prioritize and what kind of language to use. And before we get into the technical aspects of cultivation, I kind of want you to just follow me in a visualization and just appreciate the temperate seasons that we have here and our mushroom cycle across the seasons. So let's consider nature as a fungi farmer, right? So everything that we're gonna learn about cultivation is through deep observation of what nature is doing, right? So right now it is the winter, and so imagine right now there's not a lot of snow cover, but we'll talk about that. Imagine that it's spring, and it's going to start with the mushroom fruiting body. So you have the mushroom fruiting body, right? And there's a lot of rain in the spring, everything is getting green, things are growing, ideally we have enough moisture. And then comes summer, right? And summer starts to dry up the environment, and if it's really dry, you're gonna probably find a lot more wood debris on the ground because things are gonna dry out and fall off, right? And then comes fall. So imagine during fall, you have the leaves falling to the ground, you have the branches falling to the ground, you have the big, more trunks and logs falling to the ground. So you have small wood debris, you have large wood debris, you have leaf litter, right? And this is literally nature preparing the substrate on the ground. Just like we would take a bunch of hardwood and put it into a plastic bag and create that sort of artificial condition, or that's exactly what nature is doing. So you have all this wood debris over the fall to the ground, right? And imagine all the insects that are sort of burrowing under the stuff, and imagine the architecture, right, of the duff and the soil, and how, depending on the insects that are there, depending on what kind of branches fell, depending on all these variables, it's going to determine what fungi fruits, right? So just consider when we have park spaces or cemeteries cemeteries, and we're just so obsessed with like clearing up the leaves. We're really doing a disservice to ourselves by stopping the natural life cycle. We are just, so this is my way of saying, even a leaf falling to the ground has a function and purpose and is integral. It's not an inconvenience. It's without it, without decomposition of those things, there's not going to be that nutrient recycling that we need in our environment, which means the nutrients don't get recycled, the plants get weaker, the trees get weaker, there's not enough moisture in the air, not enough rain. Again, less trees, less shade. It gets hotter and hotter and hotter. So it's crucial that we understand the relationship that fungi has with our environment and the weather, and we learn to nurture it so that it can fortify our plants and trees, thereby grow bigger and stronger, and hopefully mitigate some of these climate issues, right? And so we're all a part of this. So nature as fungi farmer. Oh, one more thing. I didn't go into winter. So fall, all the wood debris falls on the ground, right? But then over the winter, you get this beautiful snow cover. And what is that? It's like moisture, hydrating that substrate, right? And then this cold cover allows for the air to be much more dense. So it's not as active. So under the snow, under the duff, the spores and the mycelium are under cooler conditions slowly growing, right? Whereas in the summer heat, it has to grow kind of vigorously and fast. It sort of can slow down that process, but it's still growing underground, preparing for spring, right? Incubating. So essentially that's what we're doing too. We're all incubating and digesting our substrates. So yeah, there is a reason for absolutely everything, and it is our duty to pay attention and to listen and to steward and help. So yes, look to nature. All right, so I'm going to take a quick breather. That was sort of the first 101 portion of mycology. So if you have any questions, this would be the time. Yes.
I really have a question, but it's about a comment that's a couple of people mentioned the taste of yeast.
Ooh, yes.
And that was in line with what you were saying, that
Totally.
the connection with fungi and yeast. So you were paying attention.
Yeah, and did you notice when you pour it out, it's got, not all teas do this, but there's like a little bit of bubbles on the edge, right? And I actually never noticed that. And when I looked at it, I said, and people were saying yeast and fermentation, I thought, well, how did that happen so quickly? How did the bubbles sort of, yeah. So I appreciate that. Any other sort of technical micro questions? Yes.
Well, not a question, but a comment. I help out with the Mycelium Park Alliance.
Yes.
And one thing that they discovered is that mushrooms will eat the excess oil.
Oh,
So like
yeah.
when you have on the river any oil that's spilled, whatever, that the mushrooms will actually eat that.
Yes.
And not take mushrooms after that, but
Exactly.
it will just clean that out. And they also noticed that some areas where they have some of the trails that have asphalt, that you can put mushrooms in there and the mushrooms will eat the asphalt.
Oh, yeah.
And I'm like, wow.
Well, that's
Mushroom burn.
their function in life, right? To break it down, for that sort of cycle of life. So yeah, they are, there is a fungi for everything. There is plastic eating fungi that eat hydrocarbons. So yeah, we're sort of just at the beginning of learning about how we use fungi to remediate our environment. I'll talk a little bit about that later. Okay, I'm gonna keep moving if that's okay. All right, so when it comes to mushroom cultivation, there are two ways that you can cultivate a mushroom. You can begin with a spore, which is sort of nature's way, Or you can actually, you don't have to change it. You can clone a mushroom. And let's say you find a mushroom that has desirable characteristics. What you can do is you can take a clean piece of tissue from that mushroom, you can expand out its mycelium, and you can clone that mushroom. I just want to talk to you about spores. So this is like a purple puffball mushroom that I use for pigment making. Calvatia cyantiformis is a scientific name. Above it are actually collections of its spores that I keep in jars that I might mix up with different solvents. And this mushroom probably dispels like millions, billions of spores. One single mushroom, right, will put out millions of spores into the environment. What's amazing about that is each single spore in the millions are all genetically unique, right? So a clone is just an extension of that one mushroom. But if you were to take a mushroom, there are so many options that you could sort of, so that's it. But the difficult thing about cultivating from spore is that they're so small, if you were to isolate it from the wild, it's very, very hard to get a clean sample of it. So it's very likely that as you're cultivating, whatever you're cultivating will get contaminated. So commercial growers tend to stick with cloning because it's just more predictable and reliable, especially for berries. Okay, and this is just some spores under the microscope. Yes.
I have a question. You mentioned contamination from your drink, you want to make sure nothing is contaminated.
Yeah.
So how does that work in nature, right?
Yeah.
Because there's all kinds of things, nothing is sterile.
Yeah, what is contamination in nature,
Yes.
right? So we're gonna focus today on outdoor cultivation techniques that don't really require sterility as much as indoor cultivation. So the only trade-off with outdoor cultivation and sort of not being as controlling about contamination and things is that you can't reliably predict your yields. When it's going to fruit, how much it's going to yield, what's kind of, impacting that. It's just, you kind of give it up to nature. There are some other ways that you can handle outdoor cultivation if you're a commercial grower. I'll talk about that a little bit later. Next slide. So if you want to collect spores, what you want to do is take a spore print. Here is an example of a spore print using these mushrooms up there. It's a cuprinoid mushroom, this is a Mycocap mushroom. This is also its cousin. But what I do is I take the mushroom, a fresh mushroom, I put it on a piece of paper. I probably look up what color it is or not if I know what the mushroom is, because if it's black spores, I use white paper. If it's white spores, I may use a black paper. If you don't know, you might just want to use some tin foil. It's another really great way to sort of capture a spore print. And it's really simple. You take the cap and you put it face down, gill side down on a piece of paper. You cover it with, say, some kind of dish, some kind of something that sort of keeps the humidity in. Just cover it with a bowl. It can be transparent, it can be opaque, doesn't matter. And before you put it in, you just want to put one drop of water to simulate rain, because that's what will trick the mushroom to release the spores onto the paper. You leave that for 6 to 24 hours. You come back to it, you remove the bowl, you remove the mushrooms, you'll see these kinds of deposits and spores. I just want to bring to your attention, this is the shaggy mane mushroom. It's the cousin to this mushroom. This is a very common mushroom that's going to pop up in your compost piles. But it does have this kind of kooky cousin, which is edible and cultivated in Asia. So again, the sort of link between wild varieties and then cultivatable varieties that are edible. And so, yeah. Next slide, please. Okay, so if you're not cultivating mushrooms starting from spore, you're probably, and you're gonna clone your mushroom using a clean piece of its tissue, there's two ways that you can expand, there's multiple ways, but these are two really common ways to expand your mycelium, right? Because you wanna expand it out so that you can inoculate other substrates. And what's amazing about mushrooms is one little piece of wedge that can overtake a whole petri dish can be used in like 10 different bags, right? So it's just this, you can use it infinite times if you keep sort of transferring and cultivating and transferring the cultures. So an agar plate, a petri dish, is basically, agar is just a seaweed derivative. It's a coagulant. And in this plate, I mix malt yeast from beer making and agar, which is that seaweed gelatin. And I mix it with water and I sterilize it in a pressure cooker. I pour it into the petri dish. And then it creates this very delicious jelly-like kind of food for the mushroom, where I take the clean piece of tissue, I put it onto the plate, and then it just loves to eat that sort of the agar plate. I just want to also bring to your attention that, you know, imagine us sort of eating a bowl of Jello or agar. The mushrooms love it too, and you know they're very similar to us. I think the more we understand them, the more we'll understand about ourselves, and vice versa. So then on the right hand side, we have liquid culture. The agar plate, of course, you don't need to have you don't need to order these petri dishes. You could do this kind of plating, you know, using a glass jar or something, right? You can modify it. You don't need to go get lab grade things. You can do it in your kitchen. Get whatever is accessible to you. Something like this. You could do this in this, is what I'm trying to say. So liquid culture is basically a nutritive solution of honey and water or sugar and water. Again, just like us, mushrooms love carbs. They love sugar, you know. They love beer, they love Jello, you know, it's all the same. So sort of like to demystify that, you know, because I know that language can get really technical, really scientific. So liquid culture, what I do here is I mix water with a little bit of honey or some kind of carb or sugar, and I again sterilize it. And I'll tell you why I sterilize it a little bit later. I then again take the tissue and I introduce it to the solution under sterile conditions, and it grows in there, right? A couple things that I want to point out about this photo. Here you see this sort of mat. This is called a pellicle. This actually happens when you kind of neglect your mushroom culture, because what you want to do is you want to keep growing it and breaking it up so that you can use it for something. But I kind of left it sitting, but it creates this beautiful pellicle, which is what these mushroom leather bags are made out of, just pure mycelium, right? Another thing to notice about this picture is at the top, you'll see some polyfill. Basically, it's a filter at the top of the jar. Remember, mushrooms need oxygen to grow just like us. No oxygen, they will die. So that filter allows for the fresh air exchange and the oxygen flow. So what is the benefit of liquid culture? Oftentimes, commercial cultivators want to use liquid culture because the materials needed are really simple and easy to find anywhere, but also because of its liquid state. When you add it to a substrate, it's able to sort of more evenly disperse through the substrate, right? Whereas this, if you're taking a wedge, it's gonna not be as evenly distributed. This also kind of, because it's evenly distributed, helps sort of jumpstart the myceliation process. It's just a little bit faster. Okay. All right, so there are about 7 stages to mushroom cultivation. And the first stage is to decide what species of fungi, what strain that you want to work with. And on the screen, I have 3 varieties of oyster mushrooms found in the wild. These are actually winter varieties. This is probably more in the summer. And you can already see from their sort of forms and formal features, you can see how this is just really kind of hardy, meaty looking and looks like it's a winter mushroom. So I'm using oysters because they are mushrooms that are easy to adapt to different substrates. I'm also signaling these wild mushrooms to say there's about 200 species of oyster mushrooms, and each of them are acclimated to different environments, right? So the pink and yellow oyster mushroom is actually a tropical variety. And the reason it's a tropical variety clearly is because it has all these bright colors. And why does it have bright colors? It's because it's gotten more sun, and so it has more pigmentation. Whereas this mushroom or this mushroom doesn't have as much pigmentation, right? It's not as tropical. Again, the formal features of a mushroom might give you a clue as to where they come from and how they survive and yeah, what those features might mean. Something that I couldn't quite look up in a scientific paper, but sort of heard from someone, the reason oyster mushrooms are able to fruit all year round is because these winter mushrooms have an antifreeze property in them. So it allows them to weather the cold weather. This is why I'm saying wherever you are based, given the substrate you have available, given the environment that you have available, you want to tailor the strain or the mushroom to that because it's just going to allow for it to grow better. So strain and species selection. I do have a catalog from Field and Forest. It's probably the most used company by commercial cultivators. It's a little bit mass produced, so I don't love promoting it because I want more diversity in the market, but it's a really reliable company. Yeah, and it's got a great catalog. They've been around for a long time. Next. Okay, so once you've decided what mushroom you're going to cultivate and you have a sense of what resources you have around you, whether it's indoor, outdoor, you want to then prepare the substrate for your mushroom. Prepare the food for your mushroom. Before the mushroom culture is introduced to its final fruiting substrate, there's an intermediary step that cultivators take, and they create what they call grain spawn. There are other kind of ways to create spawn as well. Cardboard spawn, sort of pointed out on the table. I prepared some for us to look at today. So the substrate. Why do commercial cultivators take that intermediary step for grain spawn? It's because the mushroom moves through the grain and eats it much more quickly and easily. It sort of expedites that portion of the stage of cultivation. So this is a cereal grain. Once again, it's so basically once you get the cereal grain, you want to and this goes for all substrates, you want to hydrate it with water, right? And this is usually 50-50. And again, it's actually not 50-50. What you're trying to achieve is field capacity. You're trying to hydrate your substrate, whether grain or some wood, to field capacity. And what that means is you want the water to saturate that substrate just enough, not too much, where it is too wet and might waterlog the cells of the mushroom, and not too little, that the mushroom doesn't have enough moisture to actually grow. So field capacity is basically when you prep your substrate, you don't want it soaking wet, you want to squeeze your substrate, and if just one drop or so falls, that's when you know you've reached field capacity. So yeah, if your substrate is too wet, you might find, oh, it didn't work. My mushroom just turned to mush, it oxidized and with gut to it. So grain spawn. I also want to point out to you this particular grain spawn, you can see the sort of discoloration start in the area, sort of greenish. That's contamination, and that's trichoderma, which is like a blue-green mold, and it's very, very common to come by in mushroom cultivation. I would probably not use this grain jar anymore, because if I were to use this, I'd probably grow more of this guy and not the oyster mushroom. Okay, so really quickly, cardboard spawn. Cardboard spawn, I like to promote cardboard spawn because living in New York City, you know, we have no shortage of cardboard and paper waste, right? That is our most accessible substrate that we should learn how to use. So cardboard spawn, what I do basically is I take corrugated cardboard. I also take these soup containers from, you know, recycling, and I put 1, 2, 3, 4 holes in 4 directions. So it has that fresh air flow and exchange and oxygen for the fungi. So essentially, I take the corrugated cardboard piece, I soak it in some hot water to remove any glues or pigments or inks, anything that might not be good for the mushroom to consume. And then again, I let it dry so it reaches field capacity, so it's not soaking wet and it's dry. And then what you do is, in a lasagna style, you sort of layer the corrugated sheet and the clean piece of tissue, and then you sort of build it up. And I don't know how many layers, maybe like 8 layers in here. And I can pass it around, open up the top of it. This is a really, really great way to use recycling materials. If you're doing a workshop with other, people and you want to do something hands-on, this is essentially free, right? You can put in the work to collect recycling and cardboard and stuff like that. So I'm just gonna open this up and pass it around. This has been incubating for 1 week. And I used oyster mushrooms from the food store. So I got a soup container from my recycling. I got cardboard from my recycling. I cooked up some mushrooms for dinner, and then I used the butt ends of the mushrooms to then sort of lasagna and layer it in between these hydrated cardboard sheets. And essentially, by closing it, I created this high humidity environment inside, right, this chamber. And so it's got enough humidity and moisture to keep moist and keep growing, and you can see that it always has some condensation. That's how I know that I have enough moisture in there. I'm gonna open this up and let you pass it around. Really cheap, easy way for people to expand mycelium.
What's up with temperature?
Yes, so when it comes to incubating, most substrates are spawn. So you want to keep it at a steady temperature of like 62 to 75 degrees. But with this, you don't have to be too crazy about it. I just take a cardboard box and I just put it in there and I just tuck it away. Temperature is important. You want to make sure that there's no light, because you want to trick the mushroom to think that it's underground and it's growing underground, right? Light will trigger it to think that it's above ground, and it will trigger it into a different stage. So you want ambient to no light. You want stable temperatures of 65 to 75. And humidity is not so important, because if you've done the preparation correctly, there should be enough moisture and humidity inside the chamber. Okay, so those are 2 easy. This is the DIY way. Yes.
So with this cardboard method, you don't have to worry too much about sterilization.
Thank you so much. If you could go back. So the reason why I love the cardboard, okay, so the next slide is really going to break it down for you. I pasteurize it, which means I take boiling water or really hot water and I clean the substrate. So I do sort of help it along. I sort of prep it and get rid of most of the microorganisms that might be living on it. But because the cardboard is not highly nutritive, there's not a lot of competing organisms coming to eat it. So when the substrate is really desirable and yummy and high in nutrition, that's when you have to sterilize something. Because otherwise it's just there's too many competitors coming to eat it. But when you use a substrate such as cardboard or straw that have a lot of carbon, but they're not highly nutritive, you don't have to sterilize them. You can just gently pasteurize them. That will be enough to get rid of the microorganisms that you need. So this was just washed in hot water, but I didn't start building the lasagna layers until the substrate cooled a little bit, because again, too much water, and also, let's say the lasagna, as I was building it, the cardboard was too hot, could also kill the mushroom or mycelium, right? I have to kind of look up what that temperature is, but you just want to make sure that you're allowing the substrate to cool. Okay. I guess one more tidbit, the reason I like corrugated is because it creates those channels and it's natural to kind of access that airway, that passageway. If you had cardboard that was just kind of fixed, you might not get as many airways.
I have a question about the bottles that you would use anymore. Can it be sterilized? Can it sterilize?
Oh yeah, totally. So this is sterilized because, oh man, cereal grain, that stuff is yummy and it's soft and it's easy to eat. You know, when you leave rice in the fridge, it's like, it goes bad pretty quick. So this one definitely requires sterilization. This one doesn't. This one requires pasteurization. So you have to understand your substrate and just look, is it high in nutrition, highly nutritive, or is it okay? Next slide, please. Okay. All right. So once you have taken, you've expanded your mycelium and you have expanded it a little bit of grain spawn, kind of expedited the cultivation process. What you want to do is then take the grain spawn and introduce it to your final substrate, right? This is going to be the block or the substrate the mushroom will fruit off of. Some things that you want to, so most substrates are hardwoods. Again, those are we've learned how to feed those decomposing mushrooms, woods, and wood debris and things like that. Things to be mindful of, we want to make sure that the carbon-nitrogen ratio is good. Usually, depending on the mushroom that you're growing, the nitrogen ratio is anywhere from 4 to 20% of the entire formulation. Certain mushrooms like shiitake or lion's mane, they benefit from a little bit extra nitrogen. So you just have to modify it and understand what you're working with. So that's the substrate. Usually you're using hardwoods. You want to pay attention to your carbon-nitrogen ratio. And so to add or remove nitrogen, you want to do that through supplementation, right? And so a lot of times, and we'll learn in the next presentation, one of the great, a great nitrogen supplement is actually your coffee waste or your tea waste, or maybe your mushroom wants some calcium and you want to throw your eggshells in there. You know, it's really, it's really up to you how you want to experiment with cultivation. So that's supplementation, carbon, nitrogen, and then what you want to do again is you want to sterilize your substrate if it's highly nutritive. Hardwood happens to be highly nutritive, and so it definitely requires sterilization, which means you're bringing it up to 15 psi. You're adding a lot of heat and pressure, and you are probably going to let that run for an hour, hour and a half for it to be completely sterilized. That kills off the microorganisms, and that sort of gives your mushroom the advantage once you introduce it to the substrate. when you're inoculating the substrate with your mushroom spawn or culture, oftentimes that stage requires sterility, because again, you've done all this work to sterilize it. Imagine you open up the bag, and in doing so, you again introduce more contaminants. So one way to do it, commercially, people usually use a laminar flow hood, basically fresh air being flowing out of the hood. And if you don't have a flow hood, what I use at home is called a still air box. So it's just a plastic bin. I've cut out 2 armholes. And unlike the flow hood where there's fresh, clean air flowing through, this is a still air box. And the reason the work that I do inside this box is a little bit more sterile is because the air inside that box is still. So nothing can kind of come and fall onto the plate if I clean it well. And I move very sort of very disciplinedly. Okay, just want to point out to you, once the substrate is prepared, hydrated, supplemented with nitrogen or whatever else you might need, once it's pasteurized and or sterilized and then inoculated with the mycelium or spawn, what you want to do is you want to let the mushroom substrate incubate block, right? So this is a mushroom block. This is it comes in a polypropylene bag. It usually has this white air filter, and usually if you buy the company, they come ready to fruit. It means that the mycelium has already established itself. It's run cleanly, and once the mycelium establishes itself on the substrate, it's much more resilient and can kind of ward off any competitors. Okay, so next. Okay, so there are a couple of different cultivation techniques. Today we're not going to focus on indoor techniques, so we won't focus on sterilization, but we will talk a little bit about logs and straw, and these are some of the mushrooms that you can cultivate. Next, please. Okay, so once you have introduced the mycelium to your substrate and you sort of allow it to incubate, this is a picture from a farm tour I took for far west fungi in California. This is not really the incubation stage because you can see it's already fruiting, but imagine this shade cover was probably shading it at one point. That area was probably completely dark with little to no light. And again, same thing, stable temperatures. And the incubation phase can run anywhere from 2 weeks, 4 weeks, depends on the mushrooms. Reishi might take up to 6 months, so it's really variable depending on what you're working with. So incubation is the next stage. Next. Once your substrate is fully inoculated and fully myceliated, you are ready to fruit your mushroom block. And so before you fruit it, what you want to do is you want to initiate it. You want to trigger fruiting. And how you do that, you can do that in a variety of ways. Nathan, can you go back?
Yeah.
One more. Okay, so for those of you who have seen a mushroom block in person, you'll see that usually the top of the bag is not cut open, right? It's usually cinched and closed, the air filter is there, and when it's incubating, this bag is not open, right? And why is that? When the bag is closed and it's incubating, inside the bag, it's a high CO2 environment, right? And we're literally tricking the mushroom to think that, oh, I'm underground still, right? Because underground, it's a high CO2 environment. When the mycelium overtakes the entire block and feeds to the substrate, you're ready to fruit it, right? So when you cut top of the bag and you introduce all that oxygen, all that moisture, all that fresh air, it literally is being tricked into thinking, oh, I just burst out of the ground, I'm here, right? And it's going towards the light, it's going towards oxygen. So that's essentially like how we've learned how to fruit fungi. Okay, keep going. Yep. So reishi mushroom is really unique in the initiation process. When the bag is closed in the high CO2 environment, it creates these sort of conchs. No, these are conchs. I'm sorry, I forget. But these sort of antlers. Yes. So the antlers come up in the high CO2 environment, right? And why are they kind of spiny? It's because they're looking for air, right? They're kind of reaching. They haven't fanned out. They're like moving through, right, underground. But then when I open the bag, these antlers, actually, these very antlers, fan into fungi, right? So once it has enough oxygen, it's then triggered. The initiation is triggered, and then once it has all the environmental conditions for fruiting, it fans out and sort of totally transforms, right? And this is special and specific to the reishi mushroom. We'll talk about other mushrooms in this presentation, but they all might have different needs for initiation. Some might need a cold shock, some might need a hit, whatever it is. For example, shiitake mushrooms are really activated by thunder and lightning. So yeah, they're very sentient and sensitive. Next. Okay, so once you have shocked it, you have initiated pinning, and you see primordia come out, and you're ready to fruit it. You're like, okay, I see where it's ready, to fruit. You can put it in a fruiting chamber. And I'm not going to expand much on this because this is an indoor feature. But essentially what you can do is take your block and put it in a plastic bin, or even a bag, or a humidity tent, or a Martha, or something. And essentially inside this chamber, it's high in humidity, 80 to 90% relative humidity. That gives the mushroom enough moisture to, Okay. So I also want to turn your attention to recyclables and materials that are around us. So being that I'm an artist and I like to experiment with the things that I learned in mycology, this is actually from an art show where I use experimental substrates to cultivate fungi. So this substrate actually has our lovely blue COVID masks that are like supplementing it. You can see the blue, so it's actually eating the masks. Awesome. Here I have supplemented the substrate with a dollar bill, sort of to signal the demise of our financial and social institutions. So anyway, I'm just trying to open up your mind about what you could be using, what that could mean. And I even sort of signal this very apocalyptic future where we are looking for sodas and we're taking the sugar from the soda to make liquid culture, because what else? So I successfully grew stuff in these, but not what I was hoping, not the mycelium. It turned red and I grew bacteria and stuff. But the reason it doesn't grow in soda is because the pH is not right. The environment is way too acidic. So in my experiment, what I do, this one is pure Coca-Cola, right? And this one is sort of a diluted root beer. And root beer is the one soda that has like a pH that could maybe produce life. Otherwise, it's just too acidic. Anyway, so yeah, explore and have fun. Next, please. Okay, so now I'm going to very briskly take you through 3 different outdoor cultivation techniques. The reason I have chosen outdoor cultivation techniques is because I want to teach you how to decompose a log, decompose wood chips, and yes, try to approach this subject as naturally as possible. Okay, so just go back once, you don't mind me. Yeah, and so outdoor woodland log cultivation, there are many mushrooms that you can use with this technique, but it was founded on and predominantly employs shiitake mushroom and shiitake culture. This technique is born out of Japan in the 50s or 60s, and we've sort of adopted that technique. Okay, again, I'm just gonna go through the cycles, and because we've gone through it once, I'll just speak from a little bit faster. You want to pick the right strain, right? So you want to, maybe you want a shiitake culture that's cold weather, maybe you want a shiitake culture that's warm weather, or maybe something that's both. So I probably picked something that was in the middle. And what you have is I got some hardwood spawn from field and forest with the shiitake mushroom already inoculated. I purchased what they call an inoculator, this little mechanism right here. And on the right-hand side are all the logs that I acquired for this project. So I did this over COVID at my parents' place in New Jersey. The suburbs are not the best place to cultivate fungi outdoors, but we tried and we learned. So my log yard is made of ash trees, sugar maples, and some oak varieties. And how I acquired these logs is by bothering people on Craigslist. And also some people, the ash trees I actually got because somebody had to cut down their ash trees on their property because they were infested with the wood,
Emerald ash borer.
yeah, emerald ash borer. But surprisingly, those logs fruited and they did just as well. So again, you know, who knows? Maybe that was some extra nutrition for the mushroom. So yeah, that's sort of how you go about finding logs, especially in this urban environment. Connect with arborists, connect with people on Craigslist, landowners or whatever. So yeah. That's all
In Randall's Island, we have a dump. I don't know if people could actually go in there.
yeah, picture it. Yeah. And I'll actually, okay, so go back once, I'm so sorry. And thank you for reminding me. A couple of things, you can see that the logs, they were harvested, they were cut and logged over the winter. And why do we do that? Why do we acquire our substrate and our logs over the winter? It's because hopefully, and maybe not anymore, that's when the trees are dormant, right? And the sap is not running. And the reason we want to cut it then is because the sap has antifungal properties. And if the sap was running, you could still use it, but you just would not yield as much. So, yep, those are things to remember. This is the prime time to go and acquire logs. Make sure once you acquire logs that they don't hit the ground because there will be soil fungi and other soil contaminants that will inoculate and embed themselves in the logs. And immediately after cutting the logs, you do want the logs to sit for a couple of weeks, about 3 weeks or so, because you want to make sure that, you know, whatever the log went through, it is then not reacting or sort of putting any sort of compounds into the wood that might deter the mushroom from growing. Okay. My original team, my brother and my mom, who get forced into helping me. Again, you cannot do it alone, so you definitely want to build a community around this kind of work. And what we're doing here, we have this specialized drill bit, which I have a picture for you, but it's about 1/2 inch in girth and about 1 inch deep. It's just a perfect little drill bit that you use usually with an angle grinder, but again, I'm cheap and I didn't invest in that. And you go ahead and you create these holes. Then you take the inoculator with the inoculator, you should jab it into the substrate, and then you insert it into the hole. Pretty easy. Okay, so next. I went to a workshop, instead of sort of lugging every log and stuff, this lady had this really ingenious sort of log holder, with these sort of wheels and rollers, and she was kind of easily able to just like turn it and inoculate it without having to just always pick the weight up. It's really backbreaking. One thing I missed mentioning is that the logs that you saw, they were about 40 inches in length and the girth was 4 inches. The reason you want to get logs that are 4 to 6 inches is because once they're hydrated with water and there's 100 of them, it gets really heavy and hard to carry. And imagine you have some kind of, thing you have to do with them, like soak them in water. It's just, you want to make sure that you pick a size that's manageable for you. One really amazing thing about woodland cultivation, and why it's my favorite technique, even though the work is really backbreaking, you prepare your logs. Once your logs are inoculated and incubated, say the log is 4 inches in diameter, that log will fruit for 4 years. For every inch of girth in the log is the number of years technically it will fruit. So it's really sustainable, it's really natural, it's essentially what nature does, right? Yes, next please. Okay, so once you put the spawn into the hole, what you want to do is actually cover it with wax. Here you have some cheese wax because it's food safe and things. The reason you want to do that is, again, you want to really help your mycelium compete with the elements and other pests and other organisms. It creates just sort of a protection and barrier. The other thing that it does, it also sort of covers the mycelium and helps it from drying out. It just sort of creates, protects it that way as well, keeps the moisture inside. Please. Okay, so then you want to, your logs are prepped. You want to also make sure before you inoculate your logs that there's enough moisture in them. You want to make sure that they're at least 50% full of moisture. And how do you do that? If you throw the logs in a stock tank, if they float, that means there's not enough water in them. You want to just leave it in the stock tank, and once they submerge to the ground, you know that there's some water in them. So that's sort of how you test whether a wood is ready to be used. And that's another reason why I wouldn't recommend using stuff that's already felled and already touching the ground. You're going to grow other things and not the mushroom that you want. Okay, so incubation, really important, depending on the strain, depending on your setting, depending on its food and all these other variables, is going to determine the length of your incubation. Some strains and varieties can be as quick and establish themselves within 6 months, but my log yard from inoculation to fruiting took 18 months, okay? And that's pretty common, especially with this kind of weather that we're having. A lot of people kind of get scared if they do it right, it's not gonna work, you probably did it right, it's probably just taking some time. And so what you need to learn about incubation, and this is true across all the techniques, you want to place the logs in a setting that's shaded and moist. Again, the shade, why? Because if there's too much sun or if there's too much wind, it's going to dry out your mycelium. It will die. It will not continue to grow. It will not, so that's essentially why mushrooms require shade and moisture, so that they can keep growing. Okay, almost last but not least. So once your logs are completely myceliated, and you'll kind of know because they will start showing white on the butt ends of the logs, they are ready to fruit. If you want, you could just leave them naturally, let them do their thing and let sort of the rain come when it comes. But if you're a commercial cultivator and you want a little bit more control and predictability, what you can do is you can cold shock them, which means you put cold water in a stock tank and anywhere 12 to 24 hours, you leave the logs in the cold water. And then when you take the log out from that cold water, in about 3 days to a week, you'll see it fruit. So something about cold shocking really makes the shiitake mushroom go, I have to do it now, you know, or I'm not going to be able to grow, and you know, whatever it is, it's sort of signaling to it, now's the time. This is the primordia. This is how they sort of come out at first, and then next. And then this is sort of fruiting in its glory. And if you haven't had log shiitake mushrooms, I would say they are just amazing. They're so sweet and meaty and big and beautiful. They're just unmatched in flavor and beauty. And nature truly does the best. I mean, look at this, right? So yeah, and one thing you should remember, shiitake mushrooms are really special. When they are dry in the sun, they can actually retain the vitamin D from sunlight, and you can then consume it, and you can get vitamin D from that shiitake mushroom, or that sun-dried mushroom, which is not common or easy to do with other plant varieties. Okay, next, just gonna. So, what should we do?
Yeah, so we should acknowledge the time. We had intentions of ending maybe 30 minutes ago or so. It's about 1 o'clock. I think it's up to you all where we go from here. We did want to hold some space at the end to go over to the park if people wanted to go check out the foodway and see what we're up to and kind of discuss some plans we have for this season. So we wanted to hold space and time for that. I don't know what your lives look like today. But
And
also,
I'll just say, I do have still a number of slides, but I do want to get through the main topics, just so you can understand and really tie things up. So I don't know if you're willing to give me just a little bit more time to just run through the slides, and I'll just sort of hurry it up. Okay. Thank you. All right. So another method of mushroom cultivation, is garden bed style cultivation. And I just want to point out to you, if you guys are thinking about companion planting, or you don't know where to start and you want to build up your soil, the best mushroom to go to is the Kingsbrookvaria, the wine cap mushroom. And this mushroom is actually can eat nematodes and is carnivorous, which is another thing to think about, right? We kind of are like, oh, let's be vegan, let's do this. But mushrooms, they will eat everything and anything. So yeah, the wine cap mushroom is great. It is one that you will find in the wild, but it's one that's really tenacious in the garden, and it's really wonderful for your soil. And when you are planting it with when you are inoculating your bed and planting that bed, you want to introduce plants and things that are really tall, that provide shade, and that's going to allow that mushroom to sort of establish itself better. That's really the one consideration you want to think about. It otherwise pairs really well with most things. Okay, another thing to remember, about 80% of fungi they're called arbuscular mycorrhizal fungi. About 80% of plants and trees have fungi on their roots, in and around their roots. So this is some different kinds of mycorrhizal fungi. There's arbuscular mycorrhizal fungi for the roots, There's also orchid mycorrhizal fungi, so orchid mushrooms are actually nutrified and sustained with fungal partners underground. So there's different kinds of mycorrhiza, but the ones that are best for soil you want to look up are cluster mycorrhizal fungi. So what are some of the benefits of fungi in our green spaces for plants and trees? It allows for deeper roots. It acts as a carbon filtered soil. It can protect against pests and diseases. It has different compounds, either phytochemical and electrochemical reactions. It can help retain water. It can prevent moldy roots, right? Think of how the mycelium, once established, can kind of ward off different pests, similar underground. It creates larger fruits and flowers. It efficiently extracts nutrients and also disperses nutrients in the underground web. And again, it really, I think most importantly, helps plants and trees take on more stress. Okay, so Randall's Island. Who was my Randall's Island? Shout out. Okay, so this is Sierra. She's the farm manager of the Randall's Island Garden Farm. Yeah.
I'm in the port.
Oh, okay, okay. So the wood chips that you're gonna see in the next few photos were acquired by Randall's Island Park. So you're thinking like, well, where do I get this wood chip? Where do I get this straw? Should I order it? You know, there are a network of people who are really motivated to do this kind of work. Sierra is one of them, and I just called her, I texted her, I said, hey, I need some wood chips. I take my shopping cart, roll over there, and you know, that's how I acquired it in New York City. Yes, next, please. Okay, yes, little shout out, right? So I didn't mention this. Most mushroom substrates are using hardwoods. Why aren't we using softwoods? Because in softwoods, they have resins and compounds that are antifungal, right? So that's largely why we don't use softwoods to cultivate. So I just want to complicate things and bring your attention to Mulch Fest. Well, even though Mulch Fest is really exciting and really amazing, but just consider how are we, when we introduce a softwood and mulch it and throw it around all over New York thinking we solved all our problems, do we truly understand how we're amending that soil, how that's going to change the fungal diversity, how that's going to disrupt or modify the relationships that are there. So even though it's a solution, it's something to think about. Yeah, so again, something I'm really excited about, just about 2 days ago, city finally is going to promote and do citywide composting, which is like, so I feel really excited. I feel like everything that we're doing now and this kind of news is all an effort to reimagine this landscape with more greenery.
Is it gonna be for all the boroughs?
I think so.
Because I know so far it's Queens only.
Okay.
Because I've seen compost box in my neighborhood.
Okay.
It's like a garbage bin, it's like a compost. Yes.
Yeah, so again, these are our substrates, right? Cardboard, mulch bed, city compost, right? So this is how we can decompose things in our environment. Next, please. Okay, so this is at Maggie's Magic Garden. It's the community garden that's closest to me. I went over there and I did this spiel for them, and then we also did a hands-on inoculation of one of the beds. So very quickly, we put down some cardboard, just created some kind of barrier from the soil. Next, please. We then take the wood chips from Randall's Island and we put down a nice big layer on the bottom. Okay, so what we did was really exciting. We decided to inoculate one bed with 2 different varieties. I can't remember which one was on the left or right, but on the left, let's say, or on one side, we inoculated with oyster mushrooms, okay? And on the other side, we paired it with wine cap mushrooms. And you might be thinking, well, aren't we gonna be competing for that food? Actually, no, and the reason we paired them is because oyster mushrooms are called primary decomposers. And whitecap mushrooms are secondary decomposers. What does that mean? Primary decomposers in nature are looking for those hardwoods. They come in first and they break down that hard stuff, right? Secondary decomposers follow behind the primary decomposers. And they're not interested in woods as much, but they're interested in the dung, they're interested in the compost, they're interested in the more turn stuff. So in a bed that has wood chips, compost, carbon, all those things, these two mushrooms are actually foraging for different nutrients, which allow them to exist in the same place. Next, please. They're also supplementing it with some of my coffee waste, just as an experiment. Again, we like coffee, they like coffee. Okay, next. All right, adding some paper, just however it is that you want to build up the bed. And we're doing a couple of layers. I think we ultimately did 3 solid layers. And with each layer, so again, wood chip, mushroom spawn, right? Cover with wood chip. And you never want to directly water the mycelium because it will waterlog the cells. You put another sort of layer of substrate and then you mist and water that. You don't make it soaking wet, but you moisturize and hydrate it just like we're doing with this. Essentially, it's all the same. Once you learn how to expand mycelium, I think you'll see how easy it is. Okay, next. All right, so our last layer of wood chips and mulch, and then we actually, because straw is not as nutritive, we kind of hid it underneath. We got the straw cover on top, and it just gives the wood chips a little extra shade, and because it's not as exciting to other competitors, maybe it helps. Next. It was a historically dry season last year. I'm sure if you grow plants and things, you know what I mean. So what we did is we usually, I don't want to use plastic and things, but we did put this sort of shade, a little bit of cover over it to kind of retain the moisture inside. Again, we removed it once it was really white and fluffy and things were established and it was resilient, but we were scared it would dry out in the sun. And then oysters. Next, please.
Sneha, quick question. Were you planting tall plants in this bed, like tomatoes or something?
So I don't know.
Wow.
I kind of like, I'm not, it's my closest garden, but I don't go regularly enough to sort of like help maintain the bed. I'm called on, but I just don't have that kind of time. So I'm not sure what they've done. But I imagine, like, I know people do blueberry, I know it's a raised bed, but I know people love putting blueberry bushes next to mushrooms and yeah. Okay, so another great way for you to acquire mycelium is to use spent blocks. What does that mean? That means you have that block of mushrooms and you've already fruited it. And just a little FYI is the amount of mushroom that you will yield from your block is equivalent to the dry weight of your recipe, right? So if you have 2 pounds of hardwood in there, you can expect 2 pounds of mushrooms if you've grown and done your job well. However, what do you do once you've fruited your block and it's not really growing anymore? Well, you can still use it. It probably still has a lot of life left, and you can embed that into your soils and just use it completely up. If you're looking for spent substrate blocks, New York's preeminent mushroom farm, it's an indoor farm, it's called Smallhold. They have sort of a graveyard of these spent blocks in Brooklyn. Alongside the New York Mycological Society, and I think Nathan as well is in contact with them. They are allowing the community and public to pick up these spent blocks to then supplement their soils and gardening and things. There's a lot happening, you just have to sort of get connected. Okay, so last thing I want to point out is straw and oyster cultivation. So Straw is a substrate for some reason, because the oyster is so adaptive, it does really well on straw. I can't quite name another mushroom that you would use for this technique, though people are still experimenting. So why did I want to highlight straw? Because straw is a grass, right? And it gets dried out, and it's really shelf stable, right? It can kind of live on the shelf, and it's not as nutritive as wood, so it doesn't require the sterilization. It requires pasteurization. And I also wanted to highlight straw, being that it's a grass or a plant, because the same sort of technique can probably be modified and used for invasive grasses and plants otherwise, right? And I don't know what are some of the invasives around here, but there have been some studies where people have been experimentally using Japanese knotweed, say, as the main substrate for fruiting. Mugwort, yes, you got that. Yeah, so that's why the straw is there. And so this preparation is just me actually boiling some straw in the stovetop to pasteurize it. I then, so this is it, myceliated. And then this, I'm actually just growing it on my window, by my window, yeah.
How can Phragmites work with this?
Say it again, Phragmites. I don't know what you mean by that.
The type of reed that could be maybe used in a similar way,
Oh, really?
Yeah.
Is that common around here?
unfortunately.
Yes, and they are, like, the roots are insane, and they grow
Well,
so long, it's.
So that's the that's the perfect kind of personal project. If you as a member of society, like, want to explore something, and we can all learn from you and support you, if you think like you have a patch, and yeah, could be another workshop. Okay, next. Okay, another way to sterilize your straw, or pasteurize your straw, is to use hydrated lime. For Fungus Festival, we had a station where people could inoculate some bags and containers with this pre-pasteurized straw and mix it with oyster mushroom spawn. This is Lee, somebody I work very closely with. She is adding the hydrated lime. Under this burlap bag is the straw, just to kind of contain it so it's not messy after. And what we're trying to do is we're trying to add the hydrated lime to get it past 10 pH. We want it to be as alkaline, to give it an alkaline bath. And this will get rid of a lot of the microorganisms. And once the bath is prepared, we leave the straw in there for about 24 hours. The only thing that you should be considerate of is that once you're done with the water and done with the straw, you add it to your compost or your soil, understand that it's highly alkaline, so you might have to neutralize it. Yeah, just be mindful of the pH changes of water and how you're sort of, how these things affect it. Seen from Fungus Fest, what we did, we hydrated and pasteurized the straw. We put it on this sort of, I don't know,
Like a screen.
like a screen. Yes, thank you. And we just sort of drained it. Again, not sterile, not best practice, but I think we wanted to teach the community and get people sort of hands-on. We did our best. It was an experiment. I'm not sure how sterile it was, is what I'm saying. And then we took that, we put it onto this sort of clean, prepared table, and people then with their gloves mixed it with the oyster spawn, and we just put it into something like this, a recyclable, or maybe a Ziploc bag. And I kind of like to do the recyclables because it's just readily available for us. Please. Okay, I want to share this with you. You guys are so wonderful to stay extra. I love this study. I found this study when I was researching the relationship fungi share with fire and sun and heat. for a presentation. And the reason I love this study is because it, for the first time, made it so clear to me the relationship between the sort of wild fungi and their life cycle and sort of all the things that I was learning about in cultivation and the stages and the substrate prep and things like that. So in an area by Crater Lake National Park, they studied mycorrhizal fungi after controlled burns. And what they found is the carbon nitrogen ratio of the soil really determined what mushroom was fruiting there. Also, the pH value of that soil determined what was fruiting there. Every time they burn in an area, that would reduce the carbon nitrogen ratio. It also seems like mycorrhizal fungi, things that are associated to roots, can actually withstand the heat because they're sort of shaded underground, they're hiding, rather than more ephemeral varieties that are decomposers that sort of are sort of have no chance to sort of hide under the shade of the tree because yeah, there's not enough moisture or whatever it is. Sorry. Okay. So yeah, again, carbon nitrogen ratio determined the fruiting patterns of what mushrooms grew where, and it was very consistent. So next, please. So basically, at the top, you see that when the carbon nitrogen ratio is below 26, you had more fruitings of amanitas and boletes. And then when you had the carbon nitrogen ratio above 26, you had more quaternarias or hydronati, not something I'm familiar with. Next. So you can just go back a little bit. Yes, so I think it's really, this is really exciting because the cultivation niche and the mycology niche are kind of separated. The people who do agriculture and the people who do taxonomic work, like, is kind of separated. You have like the academics that are sort of from their like soapbox, like, you know, referencing data and studying things, but they don't have that grassroots hands-on sort of experience that maybe farmers do or people working with the land. And I love this study because it sort of brought both together. And so now when you're walking in the forest and you learn to identify your wild mushroom, you may also now understand the soil composition. If you know what kind of mushroom has what kind of carbon nitrogen ratio, right? Then it can tell you something about the soil. So yeah, something to think about, something that I don't even think the Mycological Society is there yet. They're like two worlds and they sort of haven't quite. Yeah.
I know that the mushrooms that we get, the nettles compared to the cottage garden, like,
Yeah,
what are these?
exactly. Yeah, what's the substrate? You know, what is it growing on? Is it dung? Is it leaf litter? Is it wood? What's the pH of the soil? Yeah, amazing. Yeah, thanks. Okay, so I just want to leave you with a couple things about stewardship, right? This is another photo from the art project I did with the experiments of substrates. This is a Ganoderma curticii, I imagine, or sessile. Those are the two most common Ganoderma related to reishi varieties that we find in our street trees. And you see the garbage sort of in the back. And the reason I wanted to bring that to your attention is that even though this mushroom can be appreciated for its resilience and for it sort of being able to grow in this sort of harsh city environment, it's probably not a mushroom that you're going to tincture or consume, right? Because fungi bioaccumulate all the heavy metals and toxins or whatever is in your substrate, it's eating it, right? So it will then be transferred onto you, and that would not be healthy. And so you probably want to be wary about where you find your fungi. Is it like right next to the car exhaust, and the garbage, and the street corner? Well, maybe you don't want to consume that fungi. Maybe you want to culture that fungi because it's got incredible properties. Maybe you want to use that fungi to make handmade papers because that's not going to be an issue. But it's, you just want to be mindful as you're finding fungi, you do live in a very, very toxic environment, and that might be harmful to you. Next, please. Okay, so quickly, microremediation, a couple different things: biological remediation, chemical remediation, industrial remediation. As I said, there is a fungi for everything. They are natural decomposers. They have different partnerships and like eating different things. And so how can we learn more about them and use them to sort of clean and rehabilitate our waterways and our soils? And two really common, or three different methods of doing that is you can use a technique called water filtration in microremediation. So that's been used to remove E. coli from the water. This is sort of a really crude example of an experiment that essentially you have, some kind of mushroom and have the water flow through it, and in doing so, it sort of filters the water as a concept. And then bioabsorption, basically just feeding whatever substrate you have to the fungi. That could be plastic, that could be an oil spill, that could be radioactive waste, it could be, whatever. And then, of course, to combat against everything that I mentioned today is to reforest our environment. Again, we need more shade, we need more trees, we need more oxygen, we need more fungi, plants. We'll need that to save us. So. Okay, I leave you with some organizations and your existing community here in New York and also in the Bronx. And I'm sure I'm missing a lot of people, so please feel free to share with me. So number one, you definitely want to link up with the New York Mycological Society. They are a group that has been going out foraging fungi in our city parks for 60 years, and they go out every single weekend, okay? So they are a archive, they're a rich resource that is holding the information of our past and maybe our future. They have expert identifiers. So if you want to learn about wild fungi and you want to start naming them, you definitely want to link up with them because they're going to tell you what it is, and the next time you go, you'll know what it is. The James Baldwin Outdoor Learning Center was definitely our first sort of conduit into the Bronx for the New York Mycological Society. They're really excited about fungi and community building, and they represented the Bronx at the Fungus Festival, serving their free pizzas. Another thing you want to do is once you start identifying mushrooms, you want to maybe use iNaturalist. The New York Mycological Society has a group on iNaturalist. We have almost 1,600 or 2,000 members. If you post a picture, know that somebody who has a lot of experience and experts in New York are going to probably respond to you within an hour with a pretty good ID. So if you are feeling insecure, or you want to be extra safe, or if you want to do this communally, these are two things that I would say definitely you want to download and use. iNaturalist is also a really great app. Let's say you're like, I want to find this particular mushroom. Well, you can then go into the map, put in the scientific name, and maybe you'll find a patch in New York City. So that's another way for us as a community, as a society, to study the distribution of things. And of course, then Portland Park Alliance, we just partnered with them, the New York Mycological Society did, to do a research permit for 3 years in Portland Park. There's about 30 plots that are there, and the New York Mycological Society, we're then going to go together as a cohort of volunteers to identify and keep a log of the mushrooms that are growing in those test sites. So we'll learn even more about the soil and the fungi. Last but not least, Kelly Street Garden is definitely a community garden that is ahead of the curve in terms of fungal cultivation and introducing fungi to the Bronx. They been doing a lot of programming. They also have a refrigerator that's doubling as a fruiting chamber for mushrooms. And I'm gonna have an opportunity to go see that pretty soon. So you definitely want to connect with them because they already have some insight.
Who's here?
I was gonna say, I was like,
Yeah.
shout out to Renee and DK, who are both present from.
Sorry, I was like, hi, Renee, thank you for coming. Awesome. So yeah, I think that's the last slide. And thank you so much. I hope that wasn't too overwhelming. I don't know if there's really room for questions just because we've gone over so long, but I am in the city. I am accessible to you. You can TM me or email me, whatever you wish. Yeah, let me know what questions you have and I will do my best to guide you and resource you.
And yeah, thank you